To measure the aerosol size distribution, a surface mobility particle spectrometer was used for the nano-sized aerosols, configured such that it could measure from 16 to 560 nanometers. An aerodynamic particle sizer was also used, configured such that it would measure from 500 to 20,000 nanometers, or 0.5 to 20 microns. These instruments were supplied with laboratory air that was dehumidified and filtered. A series of mass flow controllers were used to control the dilution flows to the SMPS and the APS to keep the count concentrations within the measurement cell's optimum range, even at our highest concentrations. A gas sampling bag was used to dilute the e-cigarette aerosol after it was puffed by the syringe. This bag was pre-filled with 2.9 liters of HEPA-filtered lab-supplied air. A puff was conducted using a 100 cc syringe, and the e-cigarette controlled either by the laboratory power supply and a second researcher, or by this Vision spinner variable voltage battery controlled by another researcher. The puff was administered manually, with the first 20 cc's being used to establish the correct puff rate, then the next 60 cc's being puffed at 20 cc's per second. After the e-cigarette was turned off, an additional 20 mils was puffed through the device to clear any residual aerosol. This was detached from the e-cigarette and expelled to the 10 mil mark, then refilled to 100 cc's with laboratory air. This was then injected into the gas sampling bag that was pre-filled with 2.9 liters. The syringe was pumped 3 times to foster mixing within the bag. The bag was allowed to stand for 60 seconds and then was connected to the sampling port that was split flow to the APS and the SMPS, like so. The total flow to the APS is around 4.7 liters per minute. The dilution flow was around 4.5 liters per minute. The total flow by the SMPS was around 1.5 liters a minute, and the dilution flow was around 1.3 liters a minute. When the syringe injection was made into the bag, the total dilution at this point was around 270-fold. We injected into a bag instead of a rigid vessel, as we had originally designed, because the volume of the bag could be depleted without changing the concentration inside the bag.